Hello and welcome to my channel. So today's lesson, it's Heredity and Evolution from class 10 NCERT CBSE biology. So today we are going to discuss about the heredity and why is it so important. The evolution part has been deleted from the recent CBSE syllabus. So in this video also, I'll be not talking about evolution. It's only about the heredity, the Mendel's experiment, the Mendelian laws, and the monohybrid and dihybrid ratios. But before that, we should understand the heredity, or why is it important, what are the related terms that we come across, or the new terms that we come across this chapter. So let's get started with heredity and evolution. So the first thing, when we talk of the chapter Heredity and Evolution, we have to understand that heredity is something that is inherited or that is passed on from one generation to another. We have often heard everybody telling that this is in our heredity. This happens because it's in our heredity, it's in our blood, isn't it? So that is what is heredity. So that is passed on from one generation to another, and it goes on for generations. Now, goes on for generation, when I'm telling, or it is passed on for generation, it means that when under sexual reproduction, when the offspring gets the traits from that of their parents. But we also know that reproduction is of two types. It is asexual mode of reproduction and sexual mode of reproduction. So when it is the asexual mode of reproduction, that is offsprings which are arising from the asexual mode of reproduction, they hardly have any variation among themselves. They have identical genetical makeup. Now, genetical makeup means the genetic makeup or the genetic constituent of the mother amoeba to that of the daughter amoeba is exactly the similar. Now, I told you that in offsprings under sexual reproduction, I just now told you that it is inherited or it is passed on. So how do it pass on? Because the genes are passed on. So just now here I'm telling that the identical makeup, here also I'm telling it is the genes are passed on, that is the genetical constituents are passed on. Then how are these both different from each other? Just see one thing, it is told that identical genetic makeup. Identical means it is the exactly similar copy as that of the mother amoeba. But here it is passed on, but a half of it comes from the mother, another half comes from the father. So there is a mixture of both the genetic constituents. Because of this mixture, what happens? A new form arises. Always, of course, the basic remains the same. Mostly some traits which are inherited or which can be inherited are obviously passed on to the offspring, and obviously some new traits are also to be seen. Say this amoeba, or see in this one, it is the identical daughter cells that has been formed. Look wise, it is exactly similar. Not only look wise, it is not only the phenotypic but also the genotypic wise. So genetic makeup is also same. Here you can see that the offsprings are different. Different means it is quite different from each other. Easily you can identify them as different from each other, but they all are dogs. So they have the genetic constitution or the genetic makeup from that of the parents, but they have something unique also which differ from their parents. And because of that uniqueness, this arises. What is it? It is the variation arises. Now, variation is very important in the planet or for the nature, because Mother Nature always looks upon, or always it moves upon for variations. Now, why is variation important? Why is it so important that we should have some kind of variation? Let's see. Because it helps us to adapt ourselves to environmental changes. So because of this variation, you see the rabbits which are in the places where it is very cold or the temperature goes below zero, there they have fur which provides them with the warmth. But on the plains where it is not required, they are different from each other. It helps them to adapt to that of the environmental or weather conditions. It is the first step of evolution and selection. See, in order to evolve, things need to change. If that same thing continues, evolve will not happen or evolution will not happen. So in order to evolve, it has to have changes. Changes means it has to have variations. New characters are born. Obviously, based on the requirements, based on the environmental conditions, the weather conditions, new characters are born and finally brings about individuality. This is very important because in human beings, we identify each one with a separate name. But can you imagine if all of us were looking exactly similar? How much ever different names you have given? Would it be easy for us to differentiate? So variation is so important. Because of this variation, because of the genetic mixing up, it leads to individuality. Now before we start or before we go still into the chapter, let's learn about the important terms and definitions which we are going to come across this chapter. These are some terms which we have not generally come on to the other chapters of biology. So the first term is trait. It is an observable feature and characteristic or a condition in an individual and is called a trait. So it is any observable feature. Supposedly we tell that the person is very tall. So that is the trait of that person because it is a character which we can see. Because it is observable, a characteristic or condition in an individual. Characteristic means that person is tall, that is the characteristic, or a condition or whatever. So that particular thing will be called a trait. A trait can be of two types, that is acquired trait and inherited trait. See, acquired trait means due to living in a particular condition or under a particular environmental condition, you gain some traits. Like that of the Arctic Circle, Antarctica, Greenland. Those people, they can bear much more cold than us who lives on the plains. So it is very important to understand that they have acquired that particular way or their body has acquired themselves to stay in that extreme conditions. But our body is not at all acquired to that particular condition. So if we go there, we find it is extreme condition. For them, it is their environment, so they are accustomed to it. So that is an acquired trait. So it is not due to the genetic makeup. It is due to the environmental conditions. Another one is the inherited trait. Inherited trait means that all the person in a particular family, they are quite tall. That is an inherited trait. So that has been inherited from their parents. So that is due to the genetic constitution. Dominant. So when we talk of trait, there are two types of trait, acquired and inherited. And out of which the particular trait which get expressed or which we can see from outside, that trait is known as the dominant trait. So the dominant is, or it is, or the trait, the character that expresses itself in the absence or presence of a recessive trait. So it is a particular trait that gets expressed in the absence or presence of a recessive trait. So dominant is something which is, we can understand from the word that it always dominates over the others. So its dominance means it will always express itself. So the trait that gets expressed is known as the dominant trait. But it is not that both in homozygous and in heterozygous conditions. See, when we tell that a tall man is dominant over a short man, but is it so that always a person is tall? Sometimes the person is short also. So how is it? Because that particular trait is missing, that dominant trait is missing. So the trait which brings about the shortness in length or dwarfness, that trait gets expressed. So that becomes the phenotype, like that of the recessive trait. The trait which makes the person short or brings about dwarfism, that is a recessive trait. So recessive trait, it is the trait of the character that expresses itself under homozygous condition or in the absence of the dominance gene. It is either homozygous condition or in the absence of the dominance gene, because if dominance gene is there, obviously it will express itself. Then comes homozygous. Now we were just telling homozygous condition or heterozygous condition. Homozygous means that a particular allele is present at the same loci, the same allele is present at the same loci. Same allele means if it is dominant, both dominant allele are present. If it is recessive, both recessive allele are present at the same particular loci. That is, homozygous is a genetic condition where an individual inherits the same alleles of a gene from both the parents. Or when we refer to homozygous for a particular trait, it implies that both the alleles for that trait are either dominant or recessive. Next, coming to the heterozygous condition. It is a genetic condition where individual receives two different alleles from the different parents, or the alleles constituting the trait is dominant and the other one is recessive. See, hetero means it is different. Two different particular alleles are present for the same trait. Like capital T is the trait or the allele for tallness, whereas small t is the allele for dwarfness. So when capital T and small t both are present together, it is in the heterozygous condition. That is, one is dominant and the other is recessive. Whereas it is present in capital T or small t condition, it is known as the homozygous condition, because both the alleles are same. And finally, it is the gene that is the basic unit of inheritance. So the basic unit, what gets inherited from a parent to that of the children, is known as gene. When we talk of gene, first, where from did this concept come? Who laid the foundation stone for all this? It was none other than Gregor Johann Mendel, who is also known as the father of genetics. So what did he actually do, or what was his findings, or why is he known as the father of genetics? Because he was the one who told about this trait of the tall and the dwarf, or about this inheritance from that of the parents to that of the offspring. For this, he made multiple experiments using the plant Pisum sativum, or the commonly known as the garden pea, in order to show that how things were inherited from that of the parental plant and how the inheritance pattern changed due to the presence of the dominant trait or the recessive trait, either in homozygous or in heterozygous condition. So let's have a look that why did he select this particular plant for his experiment. There are so many other plants. He could have selected some other animals also, but still he went for this plant. Let's see why he did so. Because they have quite a number of observable traits, contrasting characters. Because this particular pea plant had quite a number of contrasting characters. Contrasting characters means if it is tall, the plants are tall enough. If it is dwarf, the plants are actually quite dwarf. Even the color of the flowers, some were white and some were actually full or truly pink or violet in color. Even the shape of the seed, shape of the pod, the seed were some were round, some were wrinkled, the shape of the pod, and the plant was easily available. So there was contrasting characters and the plant was easily available. So you can easily procure the plant from anywhere. It was convenient for Mendel to control the mating of the plants. So when he actually used to cross them, he was able to control the mating, or control it means he was able to do it in actual way what he wanted. The plants have short lifespan, so he could study the number of generations. So because if it was a tree, it would have been difficult for him to complete all the results within his lifetime. But because this plant has a very short lifespan, so he could do multiple of them through or within his lifespan and complete his findings. And finally, one successful cross resulted in quite a number of offspring. And successful plant or successful crossing, because if you open up a garden pea, if you open up one pod, you get at least 5 to 6 number of peas. So obviously if you cross, whatever seed it generates, you get at least 5 to 6 of them, which is good because when you need to grow them, you have ample quantity of seeds with you. So these were the reasons why he selected the garden pea plant for his experiment. These were the contrasting traits for which he had selected them. There were 7 of them, that is round and wrinkled, I told you, yellow and green color seeds, inflated, constricted, see the constrictions are there, and this is totally inflated, green and yellow, purple color flowers and white color flowers, actual termination or the location of the flower, this is terminally located, and finally tall and short are the dwarf plants. Now let's see what exactly Mendel did. What did he did for his first particular type of cross? He raised pure lines with contrasting characters to be studied. Now pure lines, the word pure line means it is the ones in which the genetic constitution is in homozygous condition. If it is capital T, both are capital T. If it is small t, both are small t. So a pure line means a condition in which the allelic pair or the contrasting characters are both in a homozygous condition. See here, capital T and small t. So both are in a pure line form. Now these two plants, he cross-pollinated with those having the respective contrasting trait to form the F1 generation. Now what he did, he actually crossed these two plants. So when he crossed these two plants, whatever he got, the F1s, because this is the first filial generation. F1 is also known as the first filial generation, which is developed by crossing of the two plants, and this particular, all of these plants in the F1 generation had capital T, small t. All of them had capital T, small t, which meant that they were in heterozygous condition. They were in heterozygous condition, but because of this dominant allele capital T, this capital T is there in each one of them. Because of that, all the plants were tall. None of them were dwarf. Though the small t was present, because it was recessive in nature, it could not express himself. The dominant expressed himself. So this is an overview of the monohybrid cross. Monohybrid, when we talk of the term monohybrid, mono means single. So first what he did, he actually took only one trait for the crossing. So he did not study for the flowers or any other characters. He just took it tall and dwarf. Because it was only one trait that was considered, so it is known as a monohybrid cross. So this is the F1, that is capital T small t, and this is the F1, that is capital T small t. Now I told you, just like the previous one, because it was capital T small t, here I have shown you with capital B small b. Okay. Now capital B small b, see, this is a violet flower. This is also a violet color flower. Now when this violet, because both were F1s and both were violet color, because violet is dominant over that of the white one. Now first this B and B combine to form capital B, because it is a homozygous condition as well as the dominant gene or allele, so it gives purple color. This is capital B and small b. So because of the presence of the dominant gene, it is also purple or violet in color. Here also it is the same thing, because of the presence of the dominant gene, it is purple in color. And here, because both the genes or both the allelic pair is small b, that is the recessive one, it expresses itself as white flower. So the phenotype what was getting expressed was white flower. So this is known as a checkerboard. When you are asked regarding to do a checkerboard and find out the number of offsprings which are having or showing one of the trait, might be a violet color, might be a tall plant, might be a round seed, which are the dominant one. So they are all three are showing the dominant trait, whereas the last one shows the recessive trait. So phenotypically, phenotypic means the one which we are able to see. So when you see the flowers, what do you see? You see that it is 3 violet is to 1 white, because you see 3 violet flowers and 1 white flowers. You don't know what is inside that. So you get a ratio of 3:1. Now we come to the genotypic ratio. Genotypic ratio means what is inside, what is the constituent of the genes or the allelic pairs inside. So what is the constituents? Here it is capital B, which is of one type. Capital B, small b, 2 are there. So it is of another type, because the genetic constitution is different, and small b again it is different from all of them. So we have 1 of capital B, 2 of capital B, small b, that is 1:2, and 1 of small b. So we get a genotypic ratio of 1:2:1. So this is the genotypic ratio and this is the phenotypic ratio. Be very clear and remember these two ratios, because these ratios are very important from the examination point of view. You will be getting question, or either you have to draw the checkerboard and to get the ratios, or simply they might ask you the ratios, because these ratios are very much in common for questions in your board exam. Now, based on this experiment that he did with one contrasting character, he formulated some laws. The laws proposed by Gregor John Mendel. So, what are the 2 laws? The first law is the law of dominance. I told you, because before explaining, while explaining, I've always told you that the dominant one expresses itself. So, that is the law of dominance. That is, in a particular combination, or even 2 individuals with contrasting trait or factors are crossed, the dominant trait is always expressed. Just as you saw in the experiment, the capital T and small t plants were crossed. What did you get? All were tall plants, but the genetic constitution was capital T, small t. So is always the dominance is expressed as the phenotype for the offspring while the recessive remains masked. So that explains the law of dominance. Second is law of segregation. Here you can see that it was capital, here it was red flowers with that of the white flowers, it was crossed. Because red capital RR is dominant over small rr, you get all red flowers in the F1 generation. F1 generation you get all red, that is capital R small r. Because capital R, that is the dominant trait, is present, it is known as, or it is the dominant which gets expressed. Law of segregation: during the formation of gamete, each of the two allelic pair for a particular trait separates from the genetic constitution. Now, when during the formation of gamete or during the formation of a gamete, each of the two allelic pair. Now, if it is only one pair of allele that you are considering, that is capital T and small t. Now, when it is during the formation of gamete, the parent having capital T gives a gamete called only capital T, and the parent having small t gives a gamete carrying small t. So each of the particular allele which constitutes a character, it separates during the formation of the gametes. See, supposedly here there are 3 pairs of chromosome. You can understand the situation. Now yellow, there are 1 pair, blue 1 pair, and green 1 pair. Now you see, when the gametes were formed, see here basically 1 pair means it is 4 strands are there. 1, 2, 3, 4. 4 strands are there. Now you see, in each of the gametes that is formed, you have 1 yellow, 1 strand of yellow, 1 strand of blue, and 1 strand of green. So in each of the gamete, from all of this, 1 strand came. Which one we don't know, but at least 1 will come from each of the particular strand. See, out of these 4 gametes formed. So out of these 4, each of them is a complete one, having 1 of yellow, 1 of blue, and 1 of green. 1 of yellow, 1 of blue, and 1 of green. So when during the formation of gametes, the allelic pairs segregate or separate from each other and forms the genetic constitution of each gamete. So these are the 2 laws postulated by Mendel after studying the monohybrid cross and declaring the results for the same. Next, we move on to the dihybrid cross. Now, dihybrid cross, when we talk of the term dihybrid, that is 2 or di comes in a question. So here we will be talking of 2 contrasting characters together. And previously we were talking of only 1 contrasting character, he will be talking of 2 contrasting characters. So here what are the contrasting characters? Round seed and yellow seeds. Round and yellow, this is wrinkled and green. So 2 characters or 2 traits we have taken, one is the round seed and wrinkled seed. So the first, when you cross, you get this condition, that is round and yellow color, because round and yellow are the dominant traits which get expressed in the first filial generation or F1 generation. When we move to the F2, we actually get 16 different types, or we can form 16 different types of gametes from F2 in F2. So what are those? If you see, that is, they are having the genetic constitution, and phenotypically if you see, that is what it looks like, we can tell the phenotypic ratio is 9:3:3:1. 9:3:3:1. This phenotypic ratio in a dihybrid cross for the F2 generation is a very important ratio from your examination point of view. Whereas the genotypic ratio, it is hardly asked, but you should also know about it. That is 1:2:2:1:1:4:1:2:1. So the genotypic ratio is quite a big one for the F2 cross, or sorry, for the dihybrid cross, but this can be calculated, but mostly you don't get questions based on this. You get only based on the phenotypic ratio, because this ratio is a huge ratio. So this is about the dihybrid cross, and based on this dihybrid cross, Mendel postulated another law, that is the law of independent assortment. What does it tell, or what does it state? During the formation of gametes, the segregation of the allelic pair is independent of the segregation of the other allelic pair controlling the trait. So when the formation of gametes, all the traits, they segregate from each other. They do not stay together, they segregate. That is, allelic pair is independent of the segregation of the other allelic pair. So how the another allelic pair is segregating, that will not matter for one of the allelic pair towards the segregation. So each of the allelic pair segregates independently of each other during the formation of gametes. So this is about the third law, or the law of independent assortment. Now coming to the another important part of heredity and evolution, that is sex determination. What decides the sex of a child? Obviously, it is the genetic constitution again, or the genetic makeup of that particular child. So let's see how does it happen, or what actually happens in the determination of sex. Now, sex determination can have two types of factors, that is non-genetic factors and genetic factors. Now, non-genetic factors generally happens for reptiles, for amphibians and them, where the formation or this determination of sex depends upon different factors, like that of the light intensity, photoperiod, temperature, nutrient availability, and the pheromones emitted by the surrounding animals and plants. So all of these actually make up to the determination of sex in that of the reptiles, like that of tortoise, snakes, etc. So if it is having very much high temperature, it might become female. If it is the temperature is a low temperature, they may become male. It is very interesting, you see, because the change in just environmental conditions can change in their sex pattern. And the next is the genetic factor. Obviously, the ones which we get to see human beings. This is common in contrast to the non-genotypic sex determination, as it establishes fertilization by genetic factors such as the chromosomes, which carry the genetic information. So if you see this is all of the chromosomes, or all the chromosomes of human beings. We human beings have 23 pair of chromosomes, out of which 22 pairs are autosomes and 1 pair is your sex chromosome. And the sex chromosome is X. X is sex chromosome for the females, and X and Y is the sex chromosome for males. So everything remains the same for a male and a female. Only this Y chromosome gets deleted or removed in a female and another X comes, whereas in a male a Y chromosome comes. This is the only difference that brings about the change in the sex of human beings. So what happens during the sex determination? It is at the time of fertilization, millions of sperms complete to fuse with the egg. Mostly the sperms carry either the X chromosome or the Y chromosome. But remember, the eggs always carry the X chromosome. And it is mostly in the ratio of 1:1. only one among the millions of sperms that are produced is able to fuse or fertilize with that of the egg. Just imagine, with the millions of sperms, only one is able to go and fertilize this egg. If the sperm carrying a Y chromosome goes and fertilizes the egg, it becomes X and Y, that is a male. If the sperm carrying a X chromosome goes and fertilizes, it becomes X and X, and that is a girl child or a female. So this explains that it is the father or the male who decides for the sex. So isn't this very interesting? So obviously it is, and genetics is still more interesting. So with this, I would like to conclude. If you have any questions, any queries regarding this chapter, Heredity and Evolution, please put in the comment section. I'll surely come back to you. In my next video, I'll be again coming up with another chapter. Till then, stay safe, stay healthy, and please do like, share, and subscribe to my channel. Take care. Bye-bye.